Okay, so welcome to the first demonstration video in the course I1 and Bioimage Analysis. Today we're going to look at EBImage and how that works. So that will allow us to do a huge variety of things, such as reading in and out standard image files, PNG, TIFF, JPEG, and so on. And that will allow us then to kind of assess more clearly different cell compartments. We can also do things like merge multi-channel images um, shown here, where we have three different channels associated with a single image that we can integrate together, like that on the right. Or indeed, we can do segmentation subject to thresholding to identify particular regions of images that are of interest. So what we're going to do first in the module, and we'll open the ORMD file to look at that further, is install a bunch of different packages. SVG or SVG is for looking at SVG files, images for image processing. Magic also is a useful tool to manipulate images. And these are for all the tasks that we're going to do, not just this one on EBImage. Colocar, as again, is for collocation analysis. Magrittr, dplyr are both for data manipulation. Reader for reading in and out files. Here to locate files, but that's not essential. DevTools allows us to open Mammuter as well later on via GitHub. Then we have ggplot2, gthemes for visualization. And then we also need to XML for our later part on the fly embryo cell movements. And then that links to CellVis3D, which you also need for that part. So what you can do is open these packages, open Biosync Manager, and open EBImage as well. And that will mean you can read in images such as the one shown here. So you can go to a website and say, call a particular package to display an image such as this simple one here. And you note that we're using the double colon here to specify that we're using this package and this function from that package, because the display function, which is a generic name, is not necessarily unique across all the different names. So now let's switch to the ORMD file where we can explore this in more detail. We can try this now on Bioconductor. If we try this function read image, that goes to the website, and then we can, if we try display on its own, we can see if that works. But if we get this error where it says use method, it is trying to use the wrong package. So what we can use instead is just put the package name at the start, and that will avoid this ambiguity such that our image can be displayed, which you see on the right. We can also read in local files. So if we merge.png file locally, we can read that image, and that will then display on our right. So you can see that image. We can also use ones that are built into the package, such as those in the image folder. We're looking at sampled PNG, and this is in the R library EBImage. And we can read that in as in one and display it. So you see there we have a bunch of parts. We can do a wide variety of things with this EBImage package. That can include taking subsets of the picture rather than taking the whole thing. So we can select coordinates as given here. So we're taking a certain portion kind of in the middle and a certain portion in the lower and mid section of this image. So we can take that first and store it in Y. And then what we're doing next is we're creating a sequence from 0.5 all the way to 5, where we're using this function gblur to take that image and to replicate it and blur it as well. Then we're combining all those 4 images together. So the output is 4, hence we get 4 images. So if we try this, function Z, and then we're going to display Z as a raster graphic. So you can see that we got blurred gradually as the image moves to the right. If we change length out to be something bigger, we'd see far more images at the end. So there we can see our numerous parts in varying degrees of blurring. So we can modify the parameters in that manner. So we can then add on top. So let's change it to say 3. So we can see it more easily. We can layer them on top of each other using this function. and then add text on top if you want the text function too. So what you can try is then putting your own text on top of the image and see what happens. We're going to ask what dimensions does this image have? So with 4 panels, it's 1536, or we know that this one is reduced panels. And we can ask then, what is Z? So Z is an object containing information about the coloring here. So it's these dimensions. So it's kind of 384 pixels high and 1152 across. And then what we can see is there's a matrix containing information about the image here as well. So that's inside Z. We can also take a file name of interest, such as parrots, and print that as a JPEG with this dimension specified and its height specified as well. So that now has taken our image and saved it out. So I've printed the file now out. We can check the file size. It's about 4950 KB. And then we can draw a histogram of the intensities in that file as well. So you can see there we have a distribution of values. So you can see a lot of them are in the middle. That's also useful then if looking at microscopy slide where a lot of it's very dark with certain things highlighted. This becomes quite useful because we see the spectrum of intensities there. We can also read in an image file known as nuke here, called nuke here, and that contains a bunch of nuclei. And then we can use print function to print that. So let's see how that works. So we're printing information about the image. It's grayscale. It contains 4 frames. So we can display all those frames together on top of each other. So you see that on the right-hand side now of our nuclei. And we can display them as a raster graphic as well. In this case, what we can see there is that we have 4 panels now. So these are the ones that were merged on top of each other. So we now see them merged together. And therefore we can subtract the maximum from each of them to identify to get the negative image as well, if you wish. So that's using the display function, which I've managed to lose. So use that display function, and it doesn't work because we need EBImage in front. So there, we can see that negative image. We can go combine nuke into frames of differing levels of changed brightness. So these functions are direct functions on the intensities that are represented by nuke. And you see that they're all changed, and we're combining this into 4 different images, and which in the image itself contained 4 at the start. So when we say to tile comb and with number of frames and line width and the foreground with the right image function at the start, what we can see there is that we have the plus 0.5 makes it much brighter. The multiplied by 3 makes it more intense. The difference is more intense. To the power of the 4.4 means that it's less clear because the intensity has been reduced. We can also set a threshold by having nuke greater than 0.3 as our threshold. So that means we turn it into 2 colors. So if we change this threshold and make it more relaxed, we can see there it's less clear. If we look at a histogram, that can also be useful in terms of distinguishing certain features. So you see there, we've got a histogram of values. So if we set the threshold actually to be much higher, let's say 0.9, we'll get a more, yeah, we can see that that's much reduced now. Yeah, and then the question is what's useful in our context. We can also crop it from the left and the bottom. So we're using this translate function, and this vector gives how much to cut from the left if positive and the bottom if negative. So if we do that, we see the bottom and left are chopped. If we do the same for the negative values and then positive values for the bottom, you can see that the cells have been cut here on the left-hand side. And on the bottom, they've been cut as well. We can also resize our image using the resize function. So that's maybe to move on to looking at flipping images. So we can use EBImage then to flip an image. So you can see that there with our cells, it's flipped upside down, and the same with flop. So flop is left, right. So you see that's gone from left to right. And then if we transpose it, we can also take our image, nuke, and transpose it as well. So that has rotated around so that it has been rearranged horizontally and vertically. We can also do a horizontal shear mapping. So here we're taking a particular angle of pi over 3, and here we're taking in a matrix where we're concatenating certain values as input. So you've got 1 minus sign of the angle, sine the angle by the dimensionality of nuke 2 over 2. So that value is going to be 510 divided by 2, and then again 0, 1, 0. So it's a matrix of modifications. So if we look at M at the end of this, we can see that contains particular values for manipulation here. So once we do that, that will create a skew in our image if we use the right function. So we can see a skewness in the image now. It's been skewed right away by the values in our matrix to perform an affine transformation on our data. So this is how we can modify images further if we so choose. Another option when we're dealing with data is to average over the pixels. And this can be sometimes be good when we have lower resolution data for which we want to segment, where we want to reduce that noise that might occur inside a cell or particle. So each of the pixels becomes the average of its nearest neighbors. This reduces noise and therefore creates some local imprecision, but it gives global character. So we're going to use filter2 and makebrush as our key functions for this. So what we're going to do is use the display function to view an image where you have applied the makebrush version where it's a Gaussian shape of a particular size and a sigma. So let's use our parts up above. So our parts we called is Z or in 1. So you see we have our parts there, and that is average them out, 1, 2, and 3. If we change sigma, that changes the standard deviation. That's relevant here. So what we can do is compensate for spatial dependencies on the background signal using this make brush function. So we're saying we're using the make brush with an argument of 5 in disk as our value, our label inside there. We're going to normalize that matrix by taking the transform of disk over to sum of disk. We're getting the frame of nuke, the first frame. We're going to filter the background based on disk and combine the original nuke first frame with the background one and the adjustment of nuke minus background where the value is greater than an intensity of 0.02. So if you apply that and then say to display tiling nuke 2, the number of frames, we can see that there at the end. Let's try that again. Okay, so what we need to do is just make sure that we're using our package for the combine function. So when we run this like this, we're then able to display nuke 2 using the tile function, specifying that there's 3 frames inside here with a certain line width. So if we run this line 159, we will then eventually see our 3 images there like that. So you can see there we have the original image, the background one, and then the almost one that's ready for segmentation, analysis of segmentation. So you can see that there's different ways to manipulate images and kind of monitor them as you go through. We could also use another image called DNA for nuclei. So we're going to read that in as our variable DNA. We'll take in the related image cells. So we've got DNA and cells. And then what we can do is display each of these. So you take our EBImage, DNA, and display that. And the same with our cells there as well. So you can see if we go back and forth, our nuclei image and then our cells. And we're going to display them there like that, except we have to use the right package, which is EBImage. So you can see there we have the nuclei and the cells displayed. And then again, we do the same for our red, green, blue image. So you see it in more detail now, the true contents of this image. So what we can do then is use the nmask_t function to apply a threshold to the image of the DNA subject to a width and height and offset to perform adaptive thresholding. So we're going to call that nmask_t, and then we're going to take nmask_f, and that's going to get the output for the fill-hill function, taking in the adaptive threshold for the DNA channel make bush to kind of smooth that out, and that will look at morphological opening and filling of holes. So that's another transform there. And then we combine nmask_t and nmask_f together in our final images using the correct function again. So there we can see the two two panels there, and you can assess yourselves if there's any differences, or if it's worth writing code to detect any differences. But certainly, so you can see certain holes on our left that are absent on our right. So that's segmentation. So what we can do then is use the dismap function to take the mask to the segmented cells properly to get the range of intensities, and then we can normalize across that range using the EBImage function. So we can see the normalized versions there, and then we can blur the cells we had before, so we do a threshold that are smoothed out and call that cmask_t. We can then take the cmask_t as our mask with other parameters to look at the cells further and call that cmask. So nmask is not declared above. So let's just look at that further. We see that's nmask_f. We run that to get cmask, and then if you apply this EBImage, we should get our image on the right. So you can see there we have the kind of background showing the areas with and without any cells across the black and white image. So that's the end of this demonstration video. Please see more for viral particle image analysis.